Yes, good. Please continue.
So I apologize for the technical problems, and thank you for the invitation. I'm the last speaker before the keynote speaker, and I will just wrap up on time. And thank you for the invitation. This is my first SCBA meeting, and so I will talk about the MRI imaging of intracranial arteries. Before I start, I would like to use this opportunity to activate our field, so what's the research in Hopkins. So basically, radiology research extended from molecular structure and functional MRI. And for MRI research, we have different areas from cardiac to neuroscience and neural function. And our mission is development of a novel MRI method and their application in medicine. We have around 70 PIs in our research divisions, and around 20 are Chinese American. So I hope next year we can see more faces from our field. So this is my our group research focus is imaging, neurovascular imaging. So this is basically what's it look like for the neurovasculature in the human. So we are focused on studying the geometry, morphology information, including lumen, also flow. Can you hear me? Yeah. Also including flow like velocity, hemodynamic parameters, and also using this information to help for clinician to diagnosis, including atherosclerosis and other vascular disease. So before we move to the intracranial MRI vessel wall or like lumen imaging, we have extensive experience in carotid imaging, because carotid is superficial. Also, it's relatively easy to assess the pathology specimen. So that is all our first start. So this is a MRA. That means your highlight the vessel. This is the bright signal is the lumen of the blood flow, and this is the vessel wall imaging. The dark is the black turns to dark. So using this image, we can highlight the wall structure. You can see this is a wall. So it has been underestimated by conventional MRA techniques. When we do the cross-sectional imaging, you see this region actually has a very big plaque. It's atherosclerosis plaque built up. When we give contrast, actually, it's nicely showing up the lipid core and the fibrous cap. The lipid core indicate the future risk. Like if you have bigger lipid core, that's more risk for future events. And this lipid core is just from the cholesterol. So that's why the lipid profile is important to monitor. So in comparing the extracranial arteries like the carotid or coronary arteries, the intracranial artery is quite different. Although they are all vessel including the 3 layers, but there's some difference, including like IEL, this internal elastic lamina. This is very thick in intracranial arteries like basal artery, but for coronary, this is just missing. Instead of that, and the EEL, that this is very thick in coronary artery, but in intracranial artery is missing or absent. So that is why there's some like aneurysm and more occur in intracranial circulation, and also the vessel remodeling is quite different compare extracranial vessel to intracranial vessels. So this is just a gross image. I put them side by side to compare the size. So this is a carotid artery. It's an endarterectomy specimen. After it's just a vascular surgeon going into the neck and remove the plaque. And this is intracranial arteries. These autopsy specimens. So that because they are very like the tortuosity or also vessel size is different, so it has some different difficulties for imaging these imaging the intracranial vessels. Here just showing like vessel ex vivo study, and we just want to like do a proof principle technique, like proof of principle to see whether we can see the layer of the vessels. This is a basilar artery, and you can see there's a 3 layers on MRI image, and this is a fibrous cap, and this is the median layer, and this is just the plaques. You can see there's a lipid core accumulated in this lesion. This is from here. So this resolution is quite high because we acquired this image at very high field. It's 17.6 Tesla. For conventional MRI, for clinical, we use in general use 3T. So this is a really high resolution, called super high resolution. So in terms of look at the intracranial vessel wall in 3T, like clinical settings. So we have to deal with several technical aspects, including resolution. We know the vessel is very small, so the principle is we need to get higher, best resolution we can get. And also for the vessel, is the round side thickness is around 0.8 millimeters. So number resolution we target around 0.5 is a 7 millimeter resolution. Because the vessel also in the brain, there's some, we know the vessel go inside the brain, that there's a blood inside the vessel, and outside has CSF. So we have to suppress blood signal and also CSF signal in order to highlight the vessel wall. For all the MRI imaging, speed is very important because especially for stroke patients, we are dealing with somebody with just most time they cannot really hold still. So we have to like acquire this image around like 5 minutes for clinical applications. This is a 2D MRI, black blood imaging. That means just the vessel, the blood, that means the blood is black, and we have particular technique to suppress blood signal and highlight this buildup. This is the lesions. This is the atherosclerosis plaques. And conventional 2D is quite tedious. And also you have, because for most patients has multiple lesions, and for these 2 slides, probably you spend 3 minutes because you have to first find this lesion, then target this lesion to perpendicular to this vessel. A lot of planning from the MRI technology side. So then in 2000, I guess 10 years before, we developed a technique called 3D vessel wall MRI. This technique is quite efficient, so we can acquire the whole grain with high resolution around 0.5 millimeter, and has strong blood suppression, and within the time around that time is around 7 minutes. So this is just showing the image. Left side is MRA. This is time flight MRA showing the vessel tree, the vessel structure. You see, this is a vessel tree. So we just do this is a sagittal view. So then we just do a vessel wall imaging along this vessel, and here is what is we acquired. Achieved 0.5 resolution vessel wall imaging. You can see that nicely the vessel lumen has been highlighted. This is lumen, this is internal carotid artery. Here is the basilar artery and vertebral arteries. And so
15 minutes left.
After we acquire this image, so we can see that, so because we highlight the wall structure, then we can just for clinician, they can do the diagnosis. Besides after beside the 0.5, around 7 minutes later, in recent years, we did a lot of optimization and current number techniques around 3.5 minutes, and resolution is also much higher. This is about highlight the basal artery as shown here. And we further go more small vessels like the Lenticulostriate vessel. This is a branch from M1. This size is very small. So, our previous work is focused on relatively large and medium size of intracranial arteries, and this is a small vessel. It's and the small vessel has a lot of indication like relate to diabetes, and also we are particularly interested in dementia. So we all know the dementia, including the vascular contribution. So that this is another contribution. We want to also look at the lenticulostriate imaging. Look at this small vessel, whether there's some plaque or some like structural change. In 2017, so in our field, we have a white paper just based on our experience, and this white paper we have contribute. We collaborate with all the neuroradiologists and also including some MRI physics. We have a recommendation. What is like the target resolution for clinical imaging and help for patient diagnosis. So we also have a couple papers just to talk about application. This is just the paper we published in 2014. Work on the stroke patients. You see there's this is vessel wall imaging. This is M1 lesion. This is atherosclerosis. So this is a plaque. And after we give contrast, you see the plaque enhanced. And then we further did association, cross-sectional association, found the enhancement actually associated with the remote downstream infarction. This is the diffusion radar image showing the stroke region. We further go.
Sorry, Dr. Qiao, we have 2 minutes left.
Yeah, so we further did application in population studies. This just showing we applied this to ARIC studies, and we did around 1980 participants, and we have prevalence reported prevalence. We found that around for the late age, around 77, 34% participant has plaques. That means if you are 70s, more like 1/3 of people has atherosclerosis plaque in the intracranial circulation. This is a kind of a population study to show the age trend between the white and the black. And we also found certain plaque, like PCA plaque, is strongly associated with MCI and dementia. And this is another imaging technique we call the green iron imaging. We have one particular method called QSM. We can look at green iron based on the green iron concentration is actually it's nicely correlated with the ex vivo, like scar tissue, rat tissue. And we apply this technique to two populations, including ARIC and the UK Biobank. We have ARIC, we have 700 participants apply this technique, acquire this image, and UK Biobank around 35,000. So we use this data to study the genetic, genetic components corresponding or increase the iron concentration. And this year, actually, UK Biobank has followed up with around 2,000 participants, and 1,000 has COVID-19 positive. So there's an open opportunity for us to study brain change or like iron concentration or neurovascular components to this COVID-19 patient. So for the future, for us, the MRI field, we always want to get super high resolution imaging. We talk about scale is different compared to all your guys, because you are doing the cellular or subcellular components, we are doing the like sub-millimeter. So that's now next two. And we also need to improve our techniques to look at the different, like make a faster or different characteristics of the tissues. And application like the artificial AI is big parts in radiology field. And there are somebody say it will replace radiologists, but not yet. So everybody right now equipped with AI techniques. So so we need to learn that. Whole department right now is doing that. And this is another group and from radiology, and we have a lot of collaborators with epidemiology, and also some physicians and yeah, and NIH grant support. And thank you so much. And this is my work, and from yeah, my group work. Yeah, thank you for your patience.
Thank you,
Thank
Dr.
you.
Qiao.
Yeah,